Hello everyone, this is Kuyalika Mukherjee, a botany postgraduate from Banaras Hindu University, and today I will discuss with you the Calvin cycle. So when we are discussing about the Calvin cycle, we should know that it was discovered by an exemplary scientist whose name was Melvin Kelvin. What he did, he used an isotopic carbon, that is carbon 14, and said that the first stable product that is formed in the Kelvin cycle is a 3 carbon compound. So the first stable compound, he said, was a 3 carbon compound. The name of the compound was 3-phosphoglyceric acid. 3-phosphoglyceric acid, which is also known as 3-PGA. This was a 3 carbon compound. So the cycle is also known as C3 cycle. The Calvin cycle or any dark reaction, it takes place. The site of the dark reaction is the stroma of the chloroplast. So what happens in the Calvin cycle? The CO2 molecule is accepted from the atmosphere and that CO2 molecule is reduced or it is assimilated to form the glucose molecule. So the CO2 molecule from the atmosphere, it is accepted and to form the glucose molecule. For this purpose, ATP and NADPH are utilized. ATP and NADPH2, these two molecules are energy intermediates. These are utilized. Where do it get from? It gets from the light reaction. The light reaction is taking place in the grana of the chloroplast and the ATP and NADPH molecules are utilized to convert the CO2 molecule and synthesize the glucose molecules. Since a biomolecule is synthesized, so this is also called as the biosynthetic phase. So the dark reaction does not directly depend on light, but it depends on the products of the light reaction, that is ATP and NADPH. If we see the Calvin cycle, three events or three phases takes place in the Calvin cycle. The first is known as carboxylation. The first is carboxylation. In carboxylation process, the CO2 molecule that is taken from the atmosphere, that is being accepted by a 5 carbon keto sugar, which is called as the RUBP molecule. RUBP, that is ribulose 1,5 bisphosphate molecule. This is a 5 carbon ketose sugar. This accepts the CO2 molecule and the first stable product of the cycle, that is 3-phosphoglyceric acid, is formed in the carboxylation. So this is the CO2 acceptor. In the second event, there are three phases. The second phase is reduction. The reduction of 3-PGA, 3-phosphoglyceric acid, is being reduced into an aldehyde molecule. In the third step, in the third event, is the regeneration. What is regenerated? The regeneration of RUBP, that is the CO2 acceptor. So RUBP molecule is regenerated from where we had started the cycle. So now we will see the three events in the C3 cycle. So as I said, the CO2 molecules that will be accepted from the atmosphere, that will be accepted by a RUBP molecule, ribulose 1,5 bisphosphate, which is a 5 carbon ketose sugar that is accepted. See, it is a CO2 acceptor and form the first stable compound, that is 3-phosphoglyceric acid, which is again a 3 carbon compound. So this is 1 carbon, this is 5 carbon. So two molecules of 3 carbon compound, that is 6 carbon. In this process, the enzyme that is used is Rubisco. The full form is ribulose bisphosphate carboxylase oxygenase, which means the active site of Rubisco can bind to both CO2 and oxygen. But normally in C3 cycle, it will bind to CO2. That means carboxylation is more preferred. But it can also bind to oxygen when there is a high temperature and high oxygen concentration. which we study in photorespiration. Now, 3-phosphoglyceric acid that is formed, it will utilize two ATP molecules to form 1,3 bis phosphoglyceric acid. Again, this is a 3-carbon compound, two molecules of 1,3 bis phosphoglyceric acid. So this is again 6 carbon. Now what happens, 1,3 bis phosphoglyceric acid will undergo a reduction process by utilizing NADPH molecules. So 2 NADPH is utilized in this process to form a aldehyde molecule called as phosphoglyceraldehyde. So here it was 6 carbon. So this is also a 3 carbon compound. So two molecules of 3 carbon compound, that is 6 carbon. After this, one of the molecules of phosphoglyceraldehyde or triose phosphate molecule is removed. One PGEL is released from the cycle to form a 5 carbon compound, that is ribulose 5 phosphate. Ribulose 5 phosphate is a 5 carbon compound. After this, the regeneration process is taking place. The ribulose 5 phosphate is regenerating by utilizing an ATP molecule. One ATP molecule is utilized in this process to regenerate the IUPAC. So what I was talking about, the three phases of this Calvin cycle. The CO2 molecule is being accepted by the RUBP to form the first stable compound, that is 3-phosphoglyceric acid. It is the carboxylation step. The second event, that is 1,3-bisphosphoglyceric acid getting reduced into phosphoglyceraldehyde. This is the reduction phase. And ribulose 5-phosphate regenerating into ribulose bisphosphate. This is the regeneration step. So now we have seen the three phases that takes place in the C3 cycle. We will now discuss the energy sheet of the C3 cycle, which is most important from the exam point of view. So as you can see in the cycle, in the reduction phase from 3-phosphoglyceraldehyde, not in the reduction, 3-phosphoglyceraldehyde converting into 1,3-bisphosphoglyceric acid, there are two ATP molecules utilized. And here in the regeneration step, one ATP molecule is utilized. So we see that three ATP molecules, and here two NADPH molecules, 3 ATP and 2 NADPH molecules, these are utilized in one Calvin cycle. We are taking one CO2 molecule, and for the synthesis of one glucose molecule, which is a 6 carbon compound, we need 6 molecules of carbon dioxide. So if we see this cycle has to repeat 6 times. So 6 Calvin cycles are required for the formation of one glucose molecule. So you will multiply it with 6. For this was for one cycle. So multiplied with 6 cycles. So 3 ATP multiplied by 6 is 18 ATP, and 2 NADPH multiplied by 6 is 12 NADPH. So, for formation of one glucose molecules, we need 18 ATP and 12 NADPH. So we can say for only one CO2 molecule will require 3 ATP and 2 NADPH, and for the entire cycle, Calvin cycle, for the entire formation of one glucose molecule, 18 ATP and 12 NADPH was required. So this was the entire C3 cycle and the energy dynamics. I hope you liked the video. Thank you.